Hello everyone, welcome back. You'll probably realize that the audio quality on this video is big poo-poo garbage. It's because, stop laughing. It's because my microphone is dead. I have killed it by overworking it and whipping it over the back, and it has been working overtime for absolutely no pay. My microphone is currently receiving CPR. I don't know if it'll make it, but now you get to hear my echo and every single bad word that's said about me in the classroom while I'm teaching. Benji? Benji holds up a screen that says insert slur. Sup?
No, never mind.
Okay. Let's do chapter 6, DNA. Biochemistry, of course. Let's talk about the structure of DNA first. Oh my god. What a marker. The structure of DNA. A lot of people don't know this, but there is a difference between something known as a nucleoside and a nucleotide. A nucleoside is the sugar ribose plus some nitrogenous base, whereas a nucleotide is a ribose plus a nitrogenous base plus any number of phosphates. This phosphate, or these phosphates, are attached to the C5 carbon, right, the 5th carbon position in the ring, and they're usually coming in 1, 2, or 3. And this would make it a nucleotide monophosphate, or diphosphate, or triphosphate, where we know this, we know ATP is adenosine triphosphate, right? So let's take a look at what this looks like. We have ribose, which comes from what pathway in the body? Hello? What pathway in the body makes ribose? Pentose phosphate pathway. This ribose looks like that. But if we're going to make DNA, what do we have to do? We have to deoxyribose. We have to take away the OH group on the 2 position. That is deoxyribose. We have the connection to a nitrogenous base, which I always draw wrong as well. And I don't want to get it wrong this time. This nitrogenous base is adenosine. Or sorry. What's it called? Adenine. Yeah. Adenine plus the ribose equals adenosine, right? So the ribose would be like that. This would be adenosine. This would be deoxyadenosine, specifically 2'-deoxyadenosine. What do we mean by this 2, 3, 5, whatever it is? The 1 is right here, because we understand that ribose had to attack the aldehyde on its side for blah, blah, whatever it is, you know that whole thing. So this is number 1. Then we go 2, 3, 4, and 5. Right, so that is the numbering situation in ribose. 2'-deoxy means we took away the oxygen from the 2 position. This is 2'-deoxyadenosine, which comes from the addition of ribose to adenine. Makes adenosine. Now, on the OH position, we are able to add phosphates and that would make 2'-deoxyadenosine triphosphate, also known as ATP. That is in triphosphate for 3-phosphate, diphosphate for 2 phosphates, monophosphate for 1. Correct? These, of course, are our high energy bonds right here inside of our phosphates. This one's going to get broken up first. You'll see this referred to by the MCAT as the gamma phosphate. Gamma phosphate gets broken up first, right? And yeah, that's that. I would be able to recognize this general structure. If you want to go the extra mile, you can upload. You can memorize upload to your brain. You can memorize the the nitrogenous bases. One thing I would know, just in terms of the nitrogenous bases, is the order in which they grow. Right? This G, have you guys come across this before? That's the smallest one. Or sorry, is it? No, sorry. My bad. Adenine is 2. This might be largest to smallest.
I think it's largest.
Yeah, so G is the largest, A, and then T is the next, and then C is the smallest. Right? Everyone says just says Gattaca, right? You guys watch House MD? Gattaca!
That's the post-MCAT adventure.
Huh?
The post-MCAT adventure.
Yeah, basically. House MD is a great show.
I'm gonna binge it afterwards.
Great show. It is definitely binge worthy. I binged it when I was 14. That and Doctor Who. But I think Doctor Who I watched like when I was 13. Oh.
Why medicine? Because I watched Doctor Who when I was 13.
No, basically. My why medicine essay sucks. I'm not gonna lie. It's just like, I don't think my life has meaning, so I'm gonna give people back life who think it has meaning. That's it. I don't think my life is worth anything, but if other people do, I'll give them their life back.
Tell me to study philosophy with it. Tell me to study philosophy.
No, actually. Let's talk about the backbone of DNA and get jiggy with it while we do it. If you have a ribose sugar, and you have a base, and then here I'm going to say that you have a continuation of this chain. We have a hydrogen here and a CH2 right here that connects to a phosphate. This phosphate will connect to another ribose sugar. I'll have one cheeseburger and what was it? The guy ordering a burger, he's like, I'll have one Whopper and one more Whoppers. Then this base will go like that, and we will have once again a connection back up to. Nope. And you'll see that the phosphate actually acts like a bridge to connect between different nucleotides, because you have phosphates on them, right? In order to build the DNA backbone, right? So what happens is that in the 5' direction, what do I mean by 5' direction? Is the direction of the 5 carbon of the ribose ring. In the 5' direction, which is going upwards, we connect up into other bases and nucleotides within the DNA backbone. But in the 3' direction, we have the OH that is making the attachment to the next one. You can see the OH here is connecting to the next nucleotide over there. Do you guys see the point? So we say we have a 5' phosphate and a 3' OH. And those two things are what's going to continue to expand on the DNA ladder. This structure is important because it gives DNA polarity. Not just polarity, but charges as well. And remember that DNA is always assembled in what direction? Huh?
5
5 to 3. So the way I drew it is actually opposite. It's supposed to go down that way.
You drew it the right way.
No, it's assembled 5 to 3. Going down this way. So the next one would be add where? At the bottom. I hate this chapter, by the way. This chapter sucks. And so does the next one. So does chapter 3. Chapter 12, now we're talking. Chapter 12 is like where I just get to do vile things to biochemistry. I get to abuse biochemistry. Yeah, it's fun. Do you guys remember that thing I did? I haven't uploaded any of the lectures we recorded from last time because like I've been spending all my time in our seminary, and our seminary Wi-Fi is like, it's good, but it's not good enough to do uploads. I think their download speeds are really good, their upload speeds are horrible. So if I wanted to upload like a 30-minute lecture, it'd probably take 5 hours, whereas normally it would take like 12 minutes. So I just haven't been able to do that, and then by the time I get home, I'm just like, I don't want to do this. So I will be doing them soon. I need to find a day where it's like, I have the motivation to do so. But, where was I going with this? Do you guys remember that diagram I made about acetyl-CoA and the 2 ways that it can go, like in terms of like the fed state, the fast state, low ATP, high ATP? Like that, I gave that discussion to one of my private students the other day, and it was like mind blowing. Like because I came up with that on the spot. I came up with that on the fly. So that was like, and I thought about it afterwards. I was like, wait, that's like actually really helpful. So I think I'm gonna make a video about that at some point. So that's what. So if you're thinking about what we're gonna do moving forward once we're done with content, that's kind of the stuff we're gonna be doing. We're talking about like specific molecules and important steps in like biochemistry and like focusing in on what you could be asked about these specific things.
But that solves a lot of my biology questions, like that.
Exactly. Let's keep going. We have other nitrogenous bases besides adenine, of course. Adenine kind of always gets the credit because of ATP. But we have adenine. You know what? I don't want to draw these. Look it up. We have adenine and guanine, which both have 2 rings, right? And those are known as the purines, right? And then we have the pyrimidines. We have cytosine, thymine, and uracil. Uracil, which is only found in RNA. What does uracil replace in RNA? Sorry? Thymine, yeah. So, we have cut the pie. And we have pure as gold. Purines are adenine and guanine. And then cytosine, uracil, and thymine are the pyrimidines. These purines have 2 rings. Pyrimidines have 1 ring. What you can do is take a little photo of whatever you have and paste it right there. Very high yield on the MCAT is that guanine is an interesting, I was about to say amino acid. Guanine is an interesting what? Base, nitrogenous base. It has a high hydrogen bond potential because of the number of nitrogens and hydrogens and oxygens that are floating around. If you look at the structure of guanine, you'll see like this double bond oxygen sticking out, and then some of the lone pair nitrogen things, and then an NH bond that comes through, whatever it is. it can make this thing known as a G quartet. We're sort of just locked into this position with 3 other guanine residues inside of the backbone of DNA. And that can be good, but it can also be no good because of the fact that you don't always want your DNA to be locked into a square. Kind of tired.
I'm gonna take a nap soon.
You're gonna take a nap soon?
Yeah.
Go for it. You guys like my sweater? Got it in New York.
It's really cute.
Keith Harington's actually my favorite artist. You guys didn't know. I really hope the audio is showing up well on this video.
I'm not gonna do it again.
I will not. If the audio doesn't show up well, too bad. You can't make me do it. Huh?
So what's so good about the G quartet?
I don't know.
Okay.
I have no idea. Guanine's kind of just like, you know what, I can, so I will. I'm sure you'll get a question about it on the MCAT. How are you going to answer it? I've got no clue. You got no idea. I don't know what to tell you. I am just messenger. Look, yeah, I can hang it, man. I feel like every time I say that, like all the brown people, like their hearts drop. They're just like, I've heard that too many times in life. It's for the white people in the back.
Yeah.
Look, yakahenge is the same thing as what? There's no white person equivalent of this.
It's just what will people
It's
say.
like-
Look, yakahenge is no. It's a lot deeper.
It
than
is
that.
a lot deeper.
It's a
but
lot
it's
deeper than what will people think. I want to be really racist right now.
Go for it. Go for it.
I'm not gonna do that.
The audio's barely in there anyways. Like you
It's
could just
okay.
be like.
No, I want to be really racist towards brown people, but
But
I
that's brown people.
But I am brown people, so
Exactly.
like
Like it's self-hate. Like that's.
yeah. Okay. I'm not gonna do it. Basically, it's the idea of like what will people think, but it's what will people think because you're such a shameful human being.
That's awesome.
Like you are such a horrible piece of garbage that like people will think awfully of our family. You know, like you've disrespected our whole bloodline. Is sort of like look yeah.
I think it's the same thing.
I think
yes. I think it's poetic.
And I mean, like white people don't really have that, but they will tell their kids to move out at like 15. So
Yeah, I was about to say we just get beat. That's fine.
at least like when, at least like when brown parents hate you, like you can still live in their house, you know.
You play with your mental health.
You go into your breakfast.
Okay, we have the Watson and Crick model. Why is it Watson and Crick? It's 'cause it's stolen. They didn't actually discover this. The double helix. Right? I remember one time I made a joke, like in my chapter 1 or chapter 2 biology lesson where we were talking about like mitosis and like DNA, whatever it may be. And I made a joke about how I was like, let's talk about some old white dudes. Because it was like Watson Crick and then like Henry, McClyde, whatever they are, whatever it is. And someone got pressed in my comment section. They were just like, there's no reason to bring race into this. I was like, buddy, I wasn't. These are all a bunch of white guys, like old white dudes. Like, that's it. And also, like, I think that every single joke that I make about science specifically has a little bit of like a historical and political background where it's like a lot of scientific discovery was made by, number 1, women, number 2, people of color, and number 3, people who are just forgotten by history. So it's kind of like unfair to just constantly be talking about these old white dudes. Do your research.
They're usually German.
The double helix, yeah, right.
Which is like a weird like.
The double helix. They talk about the sugar phosphate backbone. we have anti-parallel strands of DNA. We need to talk about this a little bit more. And we also talk about one specific thing known as complementary base pairing. If you have a ribose that is going, am I still in frame?
Yeah.
I'm just gonna write phos right here. And you have a base. Let's say that your base is A. On the opposite end of this, when this O connects this way to the next thing, and this pH connects that way to the next thing, on the opposite end of this, what you're going to have is the same thing, but dizzy, right? Upside down. Where the 5' is now here, which is going to connect off that way. And the 3' is here, which is going to connect off that way. You guys can see this is the 1 position, right? And this guy is going to have a complementary base pair to this one. Yes? That's going to be what? What's the complementary base pair to A? T. These are your bases. They are going to what type of bond across? Hydrogen bond. They are going to hydrogen bond across that. One of these runs, if you were to read it, 5' to 3', which means the other one must run 3' to 5', right? But they are both built in what direction? 5 to 3. Meaning that one of them will be built continuously, probably this one. We're on 5 to 3, 3, 3, 3, 3, 3, 3, right? And then this one's going to be built in fragments. And we'll see why in a second. It's because you can only, basically, DNA is made in the helix, correct? You unwind the helix to a certain point, right? And since you're unwinding it to a certain point, what's going to happen is that normally the DNA is like this. And what you do is you unwind the opening and it closes over here and continues. So then when you open the DNA, this one, let's say, is running from 5' to 3'. We can build this one continuously as we continue unraveling the DNA. But this one, the 5' end is over here, the 3' end is over here. We can't build this way. So we build a fragment, and then wait for this part to open up. And once it opens up enough, we build another fragment. Once this opens up enough, we build another fragment. These are known as Okazaki fragments. And they are closed using an enzyme known as DNA ligase. The ligation, right? To ligate something, to close it, right? And as we build it, those bases will naturally attract towards one another and close the bond between them and close that double helix. Do you guys remember what is the other interaction that allows the DNA double helix to close? Like, why do we get it to be so tight? No, not just that. The hydrogen bonds, yes. But what gets the DNA double helix to actually spin on itself like that? It's very specific. It doesn't come up all that often in biochemistry. Did you guys say it? Yes, that too. Okay, so that's like an extra, it's within the DNA itself. So histone proteins, yes, we'll talk about it in a little bit. But within the DNA itself, do you guys remember when we talked about pi stacking? Yeah, so it's like, it allows for that compression because what happens is the aromatic rings, the p orbitals attract one another. And allows them to completely flatten out and stack like that. What are these?
Oh, they're like those stickers I got from my nominal delivery.
That's cool.
I forgot the Gawa House one at home. I was gonna bring you that.
What base pairs each?
I would take one.
Yeah, take one. I also have the Gawa House one for you.
القلب يعرف طريقه. Interesting.
What is in there?
The heart knows the way.
Yeah, that sounds right. What are the base pairs again? I know it's AGT and what's the next one?
AGT, G to C.
G to C.
Right? Which one's more stable?
C.
G to C, why?
Hydrogen bonds.
Higher number of hydrogen bonds. How many hydrogen bonds in the G to C connection? There's 3. The A to T only has 2. It's a very important fact to know that between the adenine, and I'm going to say this very specifically, between the adenine of adenosine and the thymine of thymidine, you have 2 hydrogen bonds. 2 of them. between the guanine of guanosine and the cytidine of cytosine. Sorry, is it cytosine? The cytosine of cytidine. You have 3.
Why are you saying that?
Because when you put adenine into a nucleotide, it's now called adenosine. Between the adenine of adenosine and the thymine of thymidine, there are 2. Because I'm saying that they can do that like independently, but this is forcing them to do that in a position that they should, right? So between A and T you have 2 hydrogen bonds, and between G and C you have 3, meaning that the G to C is more stable. Now, this implicates some specific quality of DNA backbone stability. The higher your G to C content, the more stable your DNA helix, right? If the percentage of G's and C's is higher, you have higher stability. This is a very important fact to remember. As much as I hate this chapter, it is very high yield. It's a very high yield chapter.
I have a question.
Yes?
I thought DNA's polarity also had a really tight like.
It does, yeah. So the fact that you have these charges that sort of float around DNA and like allow it to do whatever it does and blah, 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 like it attracts itself, it repels itself, this, that, the other thing, it all increases in its stability because of that. Oh, I think my microphone's ready. You guys on camera no longer need to hear Benji going as he gets traumatized by what's on the board. I'm gonna pause the video for like 1 second and just merge them in post so that the audio doesn't mess up. Thank you guys for watching. I'll be back in 0.5 seconds. We're back. Okay, my microphone's alive. We have successfully defibrillated my microphone. It only took 74 tries and 17 rounds of bicarb. But you know, DNA. If you got that joke, I'm sorry that you had to go through that. The joke is that when someone undergoes like a cardiac arrest in the field, like either in the hospital or live or whatever it may be, one of the things that you do is when they have a non-shockable rhythm, there are shockable rhythms and non-shockable rhythms, which you learn in CPR classes if you've taken CPR before. One of the things you do in order to get the heart going again is you inject them with sodium bicarb. So you do rounds of CPR, you do, actually, this happens with shockable rhythms as well. If they have a non-shockable rhythm, usually you're just doing the CPR. My bad, I got mixed up in my head. But if they have a shockable rhythm, you'll deliver shocks and you'll also you'll continuously give bicarb. I think it's every like 5 minutes.
Adenosine to stop the heart, atropine to restart the heart, epi to get the heart faster. Yeah.
There you go, epi and bicarb. You cycle between them. I have to restudy my.
Oh yeah, I don't remember. Just fucking
I used to know like my heart attack protocol like the back of my hand. But to be honest, a lot of people are just like, oh, like med school is so much information. Like, how am I ever gonna remember it? Blah blah everything. Trust me, when you are in like live for your first cardiac arrest and you're watching it happen, you're gonna remember everything. When you walk out of there for like the next 6 months, you're gonna be able to tell everyone like this is exactly what happened in the room. It's very visceral. It's interesting. Like practice really is the best way to learn. Oh, okay. So we're talking about the G to C content, and this is kind of like a basic principle. The percent of adenine plus the percent of thymine plus the percent of guanine plus the percent of cytosine is going to be equal to 100%. Because there should be nothing else in the DNA, obviously. And not just that, but the percent of adenine should be equal to the percent of thymine, or adenosine to thymidine. And then the percent G should be equal to the percent C, right? And if these are off, you got a problem, right? There may have been a mutation there somewhere. If you disrupt these hydrogen bonds, right, you will naturally get a denaturation of the DNA double helix, right? And what's one way to get it to re-anneal? You can cool it down. If this freaking towel drops one more time,
Is it on a magnet?
I think it has a magnet in it, but magnets are boring.
I went off on a rant for like 10 minutes on TMSs.
What about them?
Transcranial.
Oh, yeah. Okay.
I'm learning about it now, and it blew my mind.
I need a different type of cranial stimulation. I need someone to like open up my head, pour a little bit of like detergent in there, and just rinse my brain. Just like I just need them to like put some gloves on so they don't get prion disease. Great. If I have prions in my head, guys, I'm cooked. All right. I just need someone to like cut them open, put on some gloves, and just wiggle around with my brain. Like I want them to like get in the folds and crevices. You know what I mean? Huh? No, not Dungeon. I'm thinking more like, you remember the Tide Pods that 14-year-olds used to eat and then go to the hospital? I want someone to like crack a Tide Pod over my brain.
Plastic and all.
The hydroxide in the Tide Pod might actually be good for me. You never know. We won't know until we try.
I want to become a neurosurgeon, inshallah. Do well, and want to become a neurosurgeon. I'll crack your head open.
Inshallah. Well, inshallah you become as a neurosurgeon. Inshallah you never crack my head open. You know when
put a Tide Pod in there?
you know when like, once again, brown people catching strays all day today. You know when you go up to like a brown uncle like for the first time, whatever it is, who has a funny story of I bet the women in the room. I bet you guys do. Who has a funny story about someone's reaction to when you told them you wanted to go into medicine? What do you want to tell your story?
Oh my god. So many uncles were like, why do I teach?
Why is always the thing.
Just marry a doctor?
Just marry a doctor. That's another one, right?
What about marriage your whole life? You'll be so old.
You'll be so old when you finish. Yeah, that's another one. Huh? When are you gonna have kids? That's a good one.
What about your husband?
Another one is this medicine thing bakwas, man. The doctors, they just give medicine, they just give pills. That's it, man. You want to do that your whole life?
If you want good deeds, just be a wife.
Just be a wife. Yeah, that's a good one. Where was I going with this? I forget. Hold on. Right. The one that men always get for whatever reason, they never say this to women, which is very telling. But the men always get, oh, and you're a doctor, you take care of me, yeah? Right? And it's like, and I'm just like, buddy, listen. I met you 30 seconds ago, first of all. Second of all, whenever people say it's like, oh, when you're a doctor, I hope you're my doctor, I was like, buddy, if you are in front of me when I'm a physician, that means you're 30 seconds from death's door. Do not wish that on yourself, okay? Like.
They want discounts too. I've actually done that a lot. As much as brown people love being like, oh, but you as a woman and as a wife and this and that in your household, they love their discounts.
Yes.
They love their bargains. I've found a lot of
Brown people do really love discounts.
Yeah.
Just do something easy. I remember, but then it's so funny because I don't know what it is about like brown culture and things. My parents are not like this, alhamdulillah, like at all. So if you guys didn't know, my parents have always told us not to go into medicine. Not because of any reason of like, don't do it. Just like for my sisters, like become a wife, do this, do that. My dad actually mandated that each and every single one of us at least have a doctorate level education, at least. Like he's like, I don't care what you do. I don't care if you don't work for the rest of your life. I don't care when you get married. But at some point along the way, you must at least be a master of something. And not master meaning like master's degree, meaning like you must have complete competency over some field of study. So my dad's like, yeah, you can go and you can like do whatever you want with your life. Like let's say I wanted to build a burrito company. My dad's like, yeah, you're going to be the owner of a burrito company, but you're going to have a PhD in chemistry. Like that was my dad's mandate. You had to do that. Because he just wanted, he wanted us to have knowledge for knowledge's sake. You know what I mean? Where was I going with this? So DNA, yeah.
People are gonna get so mad in the YouTube comments. They're gonna be like, "I didn't come here for a yeah." Shut up.
We just got the mic on and it's actually like the least relevant. Peter, hold on.
I'm not allowed to say anything more mean just because of my circumstances at the moment. No. I don't want you to say anything to anyone. You're gonna make them feel bad.
That's so much fun. It brings me joy.
What was I saying about this?
Medicine?
Yeah, something about my parents and medicine.
Oh, they said they will like say no, like don't do it.
Yeah, so my parents never like forced us to go into medicine, whatever it may be. But when we decided to go into medicine, my parents were just like, yeah, do whatever you want. Because at the end of the day, my parents have that faith that it's just like, you make time for what's important to you, right? Whereas I know a cardiothoracic surgeon at NYU. This guy's working 70, 75 hours a week, has 4 kids. His kids love him. The kids love the guy, right? But basically all he does is he works and he spends time at home. That's it. He works, he spends time at home, and he takes his family on vacation. And he's just like, that's my life, and I love my life, you know? So if you love your work and you love your family, hopefully you do, what's wrong with just working and going home? A lot of people are just like, oh, my life, I have to go out to the restaurant and the club and the bar and this and that, and I have to play soccer with my friends every Saturday night. It's like, dude, go home. You have a wife and children. Like, right? Yeah. Okay. Let's talk about chromosome organization. Is anyone here good at video editing? Okay. You guys are useless! Histones.
Dude, it makes it more authentic.
I can give it to my 11-year-old sister and she'll make like a CapCut video.
I definitely want the 11-year-old on my team. That would be great. Just so I can get told that I'm using child labor as well. Brown guy on YouTube, always wearing the stupid thing on his head, now using children to do his work. I don't like this guy. And he smelled bad. Speaking of, scent of the day. Again, Crystal Saffron by Mathieu. I've had this for, 2 months? Look at the bottle.
Oh, you got like a third left.
I basically bathe in this stuff. This is, out of every single fragrance I've ever owned, this is the one that I've gotten the most compliments on, by far, by a country mile. This is a very people-pleasing fragrance. Okay. I did give away some of that, though. I decanted like 30 mils of it and gave it to people. Hamza has one of them. Histones. Histones are proteins that the DNA wraps around. You guys know how an anaconda, you guys were in elementary school, you know how an anaconda sweeps around its prey and like tightens up? That's how I sort of think of the DNA in histones. What are you looking at?
Oh, we wanted to buy something.
Ah, yes. I saw the look on your face. Don't worry about it, okay? Don't worry about it. Wraps around, right? And the reason it wraps around like this is because of what? The histone proteins don't get any bright ideas. I got that on a significant discount. I paid $240 for that. No. You're probably looking at the original. You have to look at the X-Straight, the brown bottle.
It gets worse.
It gets a lot worse.
How much is it? Oh, 320 for a long night.
Let me see. Yeah, that's a small bottle.
Oh, it gets worse.
The histone protein is positive all around, right? And if the histone protein is positive, and we know the DNA has what backbone? A phosphate backbone. The phosphates in the DNA can actually attract to this histone protein and attach to it and wrap around, right? And what's interesting is that you will learn in medical school, when you look at the actual like structure of histones, that it has like all these balls and has a rod going through it, like a little protein rod, and it allows the DNA to wrap around like that. It's cool, really cool. This produces something known as a nucleosome. Guys, I think I know why like I'm so calm today. Zaid's not here. My cortisol levels are just. Oh.
He was getting jiggy and everything earlier. That's something the time of his life.
Zaid's gonna walk in the room. Oh! I need to like make a compilation of Zaid hate and just like post it on my TikTok.
Like edit up like Yusuf when Zaid and Yusuf with just like.
Have you guys seen that TikTok of Elmo trying to merge into the highway? Funniest thing I've ever seen in my life. And the other one, what's the other one where it's like the person's trying to merge, they're like driving down the lane, they're like, la 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 la, la 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 la. And then as they're screaming their lungs out while they're trying to merge the moment they're on the highway. La 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 la. I swear, the highways in New York, it feels like that. It's bad. Someone pull up that video. Okay. During DNA replication, something interesting happens. So let's talk about heterochromatin. Bye, Khadija.
Bye. I might be back.
Don't come back. Zaid's going to be back when you... No, Zaid's going to be back. So I'm just going to like start punching people randomly.
That's so nice. How long do you think you're gonna be here?
Huh?
How long do you think you're gonna be here?
I don't know, until we're done, I guess.
Okay, I'll text.
Okay. Heterochromatin and euchromatin. So heterochromatin is compact and it is wrapped around histones. And euchromatin is loose and released. So at any given time, did you find it? Yeah. Let me see. Why is Elmo such an icon? So heterochromatin is when the DNA is wrapped and compact around histone proteins, things like that. And now in order to make the DNA accessible for replication, for transcription, translation, whatever it may be, transcription, of course, because translation happens to RNA, we need to be able to unravel it from that very tightly bound histone nucleosome complex. So we unravel it into euchromatin, which is like loose, whatever it may be. And then we've talked, oh my God. We've talked about telomeres before. Those are like the little ends of DNA that like get replicated by telomerase. What are you looking at? They get replicated by telomerase and they extend the end of the DNA. And every single time you replicate, the telomeres get shorter and shorter and shorter. That's basically like your internal clock being like, hey man, you're gonna die someday. Yeah. Yeah, so that's like epigenetic control and things like that. We'll talk about that in just a second. Actually, we'll literally talk about that in just a second right now. So we talked back in biology about DNA methylation and histone acetylation. Let's talk about histone acetylation first. You guys know, because I've messed this up multiple times in my lecture series and I've finally learned the proper thing, what are the two prominent amino acids in histones? It's not histidine. It's actually lysine and arginine. Let's look at lysine. Something like that is the R group of lysine. Wake up. When you have that amine terminal of lysine, right, this is the part that's basically attracting the phosphate, correct? Make sense? Imagine if we took this, right, I'm going to draw it out explicitly. Imagine that we took this positive thing and we did this. This group is known as an acetyl group because acetyl means 2 carbons, acyl means any number of carbons, right? Do you guys remember from chapter 11, acyl-CoA, right? Meaning any number of carbons with that double bond O-CoA at the end. I have a tiny spoon in my pocket today. So similar to the rice grain that I pulled out during the metabolism lecture, I have this tiny spoon. I thought it was Zaid. I was going to flip.
That's what I found, actually.
Oh my God.
I had to go back. I saw him walking down the hall. I was like, no way.
Oh my God. How you doing, man?
Tired.
Oh, he took my C and everything. Perfect swap out. Okay, bye for real now.
Guys, Zaid is here. That means we are going to stop having fun.
We're going to start rage baiting.
You can see that this acetyl group right here, 2 carbons with that double bond O on it, is attaching to the nitrogen terminal that was once positive. Now, by attaching, what happens is we get a neutralization of that positive charge. What does that do to the DNA histone interaction? It actually disrupts it. It disrupts the interaction between the negative backbone phosphate of the DNA, the negative phosphate backbone of the DNA, and the positive of the histone. So now, not only that, but we see over here we're going to actually have resonance. So there's like an electron net movement of electrons across that terminal. It's going to repel the phosphate of the DNA. And if you do that to enough of the histone is basically going to spit the DNA back out and make it more available. So that is the transition from heterochromatin to euchromatin, making the DNA more accessible and open in order for us to do whatever we need to do. DNA methylation, specifically, this relates to just DNA structure in general. DNA methylation specifically relates to transcription. Imagine you have an open DNA segment that you are trying to read with an enzyme in order to copy it, RNA polymerase, right? And all of a sudden, someone throws a branch right here. That's the methyl group, let's say. As you're reading, you hit the branch and you can't move forward. So that actually terminates the sequence and stops you from moving forward in order to go and transcribe the DNA into mRNA. So this actually leads to a decrease in protein synthesis overall, because if you don't get transcription, you can't get translation. So those 2 ways that we modify the genetic activity, DNA.
So does histone acetylation just unwind the DNA, whereas DNA methylation actually stops?
Yeah, exactly. So histone acetylation will unwind the DNA, take it off of the histones, right? And then DNA methylation will block transcription.
Is there a process like that that gets you from heterochromatin to euchromatin?
To go from heterochromatin to euchromatin, I assume it's mostly histone acetylation. Mostly, I assume. I don't know. I'm not a biochemist. Might as well be at this point. Actually, I don't want to. I have to take like analytical and physical chemistry, and I don't want to do that. Let's talk about DNA replication. There was a point in time where I wanted to be a chemistry major. I really, really, really love chemistry, but I am academically shot at the moment. I do not want to study anything other than what I study now. DNA replication. This is going to get testy. No pun intended. We have a set of enzymes or proteins that assist with DNA polymerization. These are known as the replosome. And DNA unwinds at something known as the origin of replication. Which produces a replication fork, which we'll look at in just a second. How many origins of replication do bacteria have? 1. What about humans? Many. You have a bunch, right? Meaning the DNA can unwind and break and continue to be synthesized from multiple points at the same time, right? Which makes sense because we have chromosomes, not plasmid, right? We have multiple chromosomes, places where the DNA can be broken up. We also have a much larger genetic sequence, much larger genetic code than bacteria do. Make sense? Very good. Some important enzymes. Helicase will unwind the DNA. You have topoisomerase, which decreases torque on the DNA helix during unwinding to stabilize. Because what you're doing is you're unwinding one portion of it, which is actually twisting it in a positive direction. And by twisting it that way and unwinding it, you're exerting a force on the replication fork where everything is still wound. To undo that force, you have a counterforce that is implicated by topoisomerase to take away that torque, right? So it decreases the overall torque on the DNA helix at a molecular level. This is an unbelievable, ridiculous, realization when you see how complicated that is. And the fact that it happens on a molecular scale. DNA polymerase, or DNA polymerases. They will read DNA, or read the template strand, 3' to 5', and synthesize 5' to 3'. This whole discussion of template strand, coding strand, non-coding strand, blah, 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 this is that. What is my recommendation on how to approach this? I have absolutely no idea. It's been 4.5 years since I've taken this exam. I didn't even know the proper way to approach this when I took the exam. Good luck. We have 2 strands. We have the leading strand and the lagging strand. The leading strand is synthesized continuously from 5' to 3'. The lagging strand is synthesized in the opposite direction of the replication fork. How far is MOTW in Plano from here? 15 minutes? I was thinking of doing their big room for our social after Ramadan. You know that big room on the side?
Yeah, that'd be a good
Yeah. And I feel like they're kind of like social media forward and everything, so it'd be nice.
They're also really big.
Yeah, they're huge. That's why. I went there the other night with one of my buddies, and I really enjoyed the space.
Because they host Mandy nights before Eid and stuff like that.
Yeah.
You just use the second half.
Not just that, but it's just I wanted a gender separate event, so it'd be nice to just have 2 halves of the room be 2 different sides. Okay, let's take a look at that for a moment. This is what the DNA normally looks like. Yes, that's a real photo that I took when I went inside the cell, into the nucleus, and towards the DNA. I took that picture. I know, thank you. Then when the DNA begins to unwind, you can imagine that removing that, there's going to be a lot of torque right here. There's going to be a lot of pressure right there. So what happens is that since you're unwinding that way, the topoisomerase unwinds the opposite way right here to decrease the torque on that part of the DNA. This is the 5' end. This is the 3' end. This is the other 5' end. This is the 3' end. So we can identify that this would be the lagging strand, and this would be the leading strand. Because why? What's going to happen is that helicase is going to continue to unwind the DNA, right? Unzip your genes as you go down the. You guys don't remember that joke? You know. It's going to continue to unwind going that way. And what's going to happen is that DNA polymerases are going to produce DNA in this direction. 5' to 3', towards the replication fork. So as the replication fork opens and more of the DNA gets exposed, you get more building in that direction. But as things open here, what's going to happen is we built this way until we got to the end. Then when we got to the end, more of this was open. So then we come back here and we build this way until we got to the end, which is where we left off before. And there's a little tiny gap in between because we can't connect the two of them using DNA polymerase. Then we need to wait for this to open more. So this is why it's lagging. You build it, you wait, you build it, you wait, you build it, you wait. And then with those gaps right here, these get filled in by DNA ligase. Drops a little closing fragment right there. These fragments are known as Okazaki fragments. Okazaki fragments. What are the nucleotides used in DNA synthesis? Interesting enough, it's actually the nucleotide triphosphates. What you do is you add the ATP, the GTP, the CTP, the TTP, and you kick off two of the phosphates, which helps because, of course, the synthesis and creation of those hydrogen bonds and bringing all those charges together and stabilizing everything and curling it back up, it's a very energy-demanding process. So by breaking off those phosphates and kicking them to the curb, you actually get that release of energy from the phosphorine hydride bond within the ATP, GTP, CTP, UTP, TTP backbone, and then that allows you to create the DNA double helix. You look like you're having the time of your life. And what enzyme do we use to replicate the ends of the chromosomes, everyone? The ends of the chromosomes. Telomerase.
You don't replicate them.
Yeah, you do. How do you make telomeres?
But they die every time.
They don't die. A little bit of it dies. Okay, pause. There's a little bit of confusion here. You have a chromosome. Notice, I didn't draw a second strand. Chromosomes don't always look like that. They look like that after the S phase, right? But I guess since we're doing replication, why not? At the tops of these chromosomes, you have telomeres, which are non-coding regions in the DNA. When you are replicating the DNA from, actually, it's a good example. When you're replicating the DNA from this, oh God, I think that person has cancer now. When you're replicating the DNA from this, wow, they really have cancer now. It's not even the same color. When you're replicating the DNA from this and it splits off into two distinct synthesized portions, which gives you that sort of double stranding, what happens to these parts? We replicate up to here, and then telomerase, if we were to zoom in on this, right, like go on your iPad, just zoom in, right? If we were to zoom in on that, what's going to happen is that telomerase will build, but telomerase doesn't have the power to build every single one, so you just shave off the tiniest bit. Every single time the DNA replicates, you shave off a little bit, and a little bit, and a little bit, and a little bit, and the telomere gets shorter and shorter and shorter and shorter until your time comes to pass. What's the ayah in the Quran? وَعْبُدْ رَبَّكَ حَتَّى I think it's آتِيَكَ الْيَقِينَ. Yaqeen. I have to look it up. I'm going to look it up. It's actually really cool, like a little Arabic thing.
I thought I just got lost in biochem there.
Yeah. وَعْبُدْ رَبَّكَ حَتَّى يَأْتِيَكَ الْيَقِينَ. Arabic is cool. I'm going to show you guys something real quick. In the Quran, it says, وَعْبُدْ رَبَّكَ حَتَّى يَأْتِيَكَ الْيَقِينَ. If only my English writing was that good. And it says, and worship your Lord, وَعْبُدْ رَبَّكَ حَتَّى until يَأْتِيَكَ comes to you al-yaqeen, the certainty. Yaqeen is like a certain, decisive, like unavoidable, inevitable, infinite truth. And al-yaqeen here, the truth that they're talking about, so like obey your Lord until the truth comes to you. The certainty comes to you. What is this certainty they're talking about? They're talking about death. Obey your Lord until the exact precise moment that you die. Because there's only one, like, in Islam, there's this big idea that, like, there's not only one truth, but there is one thing that will happen to every single person, and it's death. So it's saying, since you know that's going to happen, since we're absolutely certain that that's going to happen, وَعْبُدْ رَبَّكَ حَتَّى يَأْتِيَكَ الْيَقِينَ. There's, like, another reference to that where it says, لا تموتن إلا وأنتم مسلمون, right? Which means لا تموتن, really, like the noon there, like the little extra letter there is for emphasis. It's like, and certainly don't perish except in a state of full submission to your Lord. Like, definitely don't do that. And now the question becomes, like, how does any single person know when they're going to die? You don't, right? So the implication there is that since you don't know when you're going to die, and you have to die in submission to your Lord, be in submission to your Lord always. Always be in a state of submission to God. Islam is actually a lot more logical and, like, conclusive than people think it is. There's a lot of logic in our religion. Okay. That's really like all of the enzymes you guys would need to know about. There is one more, like, that we didn't talk about, DNA primase, right? It lays down the primer in order to begin, like, DNA transcription, things like that. Just remember that the DNA primer that's put down is actually made of RNA. Why? I don't know. I have no idea.
I read somewhere you can outgrow HSV essentially due to telomere shaving off all the time because that's where it's attached to.
Interesting. That's cool.
In theory, you can outgrow herpes.
Wow.
I don't know if it's true. I read it.
Outgrowing herpes through telomere activity. That's interesting. Guys, please, no one become patient zero for that one. Don't do that. We talk about DNA repair. This really is just like P53. They'll ask you about that every now and again on the MCAT, where it's just like, oh, P53 recognizes, changes in DNA. Retinoblastoma is a similar one, where that's a type of cancer, but it's named after, like the gene is named after the cancer, right? And then oncogenes are mutated genes that cause cancer, and primarily they encode cell cycle related proteins. That makes sense, right? Because if you have changes in the cell cycle gets damaged, you actually have a change in the way that the cell cycle runs, and you can get an aberrance, an overactivity of the cell cycle, so you can get rapid division without control, correct? DNA proof reading. This is a really difficult part of the chapter. I also never, ever fully dedicated this to memory. It's the whole mismatch repair, nucleotide excision repair. Take a look at that whenever you have a chance.
You talked about cancer. It's essentially the phases, but apoptosis is not there.
Yeah. So there's actually a very, very, very interesting article that you guys should read. It's an old article. I think it's from the 80s, maybe. You guys should all read it. It's awesome. It is called you can look it up now. Wow. It's called the 6 Hallmarks of Cancer. It is a article that details the 6 changes that take place genetically in order to give cancer what it does. And basically, it has the avoidance of apoptosis, formation of its own blood vessels, removal of cellular anchors, ignoring apoptotic signals inside of the body. So like it inactivates like the back pathway, how it inactivates the cellular anchoring, how it uses VEGF to its ability to bring in more blood so that it can eat more glucose and grow more rapidly. This very groundbreaking and striking paper. 6 Hallmarks of Cancer. It's incredible, right? Incredible paper. And it's so well known that it's like completely free now. You can find a PDF of it anywhere. I actually had like one of my favorite things to learn in medical school was cancer, because we had not because of it's exciting or anything. It's very sad, but molecularly, at a molecular level, cancer is a very interesting to study if you have a good professor. We had a fantastic professor, very, very good professor. I think her name was Dr. Blaine. Great, very intelligent woman. The rest of the chapter speaks about DNA libraries and DNA recombinant technologies and blah, blah, and this and that. I'm really bad at all that stuff. I'm not going to lie to you. If I tried to explain it to you, I'd probably confuse you more. I'm not going to lie. You should go watch Dr. Iman's video on that. She got her PhD recently, if you guys didn't know. So she's no longer Professor Iman. She's Dr. Iman. She got her PhD last year, in the summer. She actually teaches at UT Austin, I believe. She and her husband live in Austin. If you guys didn't know, she's a professor there. She's a professor of physical chemistry and general chemistry at UT Austin. You guys didn't know that?
No.
Yeah. No, she's a full-time professor. Like she's a baller. Yeah. So shout out to Aman. I mean, definitely go watch her video. I recommend them all the time. A lot of them are probably better than mine. Like for some of the topics I'm bad at. Like my chapter 8 video for general chemistry is nowhere to be found. Go watch Professor Aman's video. Physics, she has all the physics stuff on her channel. She's great. I saw this video online on TikTok of people can be comparing. It's what should you use Iman or Yusuf for this or that, blah, 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 whatever it is. And this is I'll give you the general consensus. The general consensus, these are not my own words. The general consensus is that it's a lot easier to digest the general science information from her, meaning the general biology, the general chemistry. But when it comes to the orgo and the biochem, I just go ham. I go absolutely full throttle into exactly what you need to know. And that makes sense because you guys remember that even when we were doing biology, I was throwing biochem into there. I was sort of referencing it all the time. If you're someone who really just needs the information laid out in front of you and you need to explain it in a way that is very digestible and easy to follow and you need it done stepwise, she's definitely your person. At least that's what public consensus says. If you're someone who really cares about application, and it just shows in the different ways that we teach, these are just our different personalities, I'm much more application forward. Let's move on to the next chapter. Chapter 7. No, it's fine. This is just going to be the laughing stock video of my channel because this is chapter 6, 7. RNA. I also hate myself, don't worry. That's a stupid R. What do you call a nurse in Canada? An RNA.
Oh god.
Let's go back on a topic that we know very well and have beaten to death. The central dogma. We have DNA turning into mRNA, turning into proteins. This process is known as transcription. This is translation. And this is replication. This is the basic way that your body functions. Messenger RNA carries the code for a specific amino acid sequence, codon by codon. And it carries this to the ribosome in order for a protein to be produced. In order for the mRNA to be translated into the protein at the ribosome, we need the action of tRNA, or transfer RNA, which carries an anticodon to the mRNA to match up to it, and it leads to the depositing of an amino acid in response to this binding. So if you look at the tRNA loop, it's actually drawn in a very interesting way. That's kind of what tRNA looks like. And there's 3 little nucleotides right here, which are going to comprise the anticodon of whatever mRNA you bring in over here. So when those two attach through hydrogen bonding, this amino acid gets kicked off and added to a chain that is going to continue to grow across the ribosome. Makes sense? Charge your phone, I'm going to need it. Computer. Codons have something known as the wobble phenomenon. Meaning that if you have a codon like UUG and UUA, the UUG will make leucine, and the UUA will also make leucine. Why is this the case? It's a protection against mutation. In case anything goes wrong or some sort of transcription or translation or blah, 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 or this or that goes wrong, then you have a ability to sort of ward off from that change, correct? There is an innate ability to ward off from mutation damage that occurs at the point that you have codon recognition. Does that make sense? Yeah? I know you just walked in, but does that make sense? And then we have stop codons. Stop codons, you should know, are UAA, UAG, UGA. Let me draw it a different way. UGA and UAG. People remember this by saying, you are annoying, you go away, and you are gone. You know, stop. All 3 of these are stop codons, right? If, pop quiz, if not a single one of you gets this right, I'm leaving. Okay? we're done teaching for the day. I'm going home. If you have a mutation in DNA that causes a premature stop codon inside of the mRNA, what is the name of that mutation? Huh? A nonsense mutation. Very good. It's called a nonsense mutation because the protein doesn't make sense. It's truncated. It's too short. Can't do anything with it. And do you see how easy that would be if this U just turned into an A? Done. Stop codon. This is why DNA synthesis and DNA replication, this and that, whatever, it's so conserved. It's so specific. It's so tightly regulated. Make sense? You look very happy in this photo.
Because it was a repeat, and my whole picture was bad, so I knew exactly what I was doing. I had like the goggle on, and I knew exactly what I did with my face, because it was a repeat.
Crazy. All right. Talk about mutations, gently wobble. We talked about mutations before. Let's just quickly go over it one more time. You can feel the Ramadan in the air. You can feel it. Huh?
What is it called, hunger?
Can you tell that we're like safety? No, you guys should.
I'm actually currently I've unlocked my first Ramadan superpower. You guys don't know it. I'm currently floating 6 millimeters off the floor.
You're hovering.
I'm levitating.
Comes after the first 10 days of
Yeah. So the first 10 days you unlock one power, and then the next 10 you unlock another.
First racist joke of the day, you're
By the 29th night, you will actually see me in the sky over Carrollton, spinning. If you guys see a shooting star on the 29th night of Ramadan, that's not me. That's me jetpacking my way back to New York City.
They're like just straight up.
Mutations. We have silent mutations, missense mutations, nonsense mutations, and frame shift mutations. Silent mutations are the ones that implicate the wobble phenomenon. So this is a mutation on a codon or a specific nucleotide base of the base of the codon that does not implicate any change inside of the mRNA itself or the protein itself. Meaning that the amino acid that's produced from a silent mutation is the same as if there were no mutation at all. A missense mutation just means you have the wrong amino acid. This is common like the V6E mutation. I believe that is the hallmark of sickle cell. Did I teach you guys how to read mutation codes? How do you read that code? They don't teach it in the book, but it's pretty straightforward. How do you read that code? What does V stand for? Valine. 6 means 6th position. And E is? Huh? Glutamic acid, right? Sickle cell, yeah. So the valine in the 6th position was swapped for glutamic acid. That's a big change. Because sometimes there are some missense mutations that are light, and there's some missense mutations that are quote-unquote heavy. If you swap the valine for something like leucine, would that really make all that much of a difference? Probably not. Because they're both aliphatic, they're both like non-polar, whatever it may be. Their R groups are kind of the same size. You're swapping something that is non-polar aliphatic for something that is acidic. That's a big change. It's a huge change.
I feel like I've seen that question multiple times.
Yeah.
Which swapping of which amino acid?
Yeah, so that's actually a pretty high yield question that you get a lot. Like which of these, they'll give you a bunch of like codes, V6E or V6L or whatever, blah, blah, whatever it may be. And they'll ask you which of these will implicate the greatest change in the backbone. And that can get testy because like, you might have like an aliphatic swap for an aromatic, and then an aliphatic swap for a basic, and you have to understand that the basic is a bigger change because you're implicating charges. You know what I mean? Does that make sense? Yeah. How's the UWorld going, guys? You have a question about? You're just asking now. Okay.
Oh, okay. Is your UWorld percentage supposed to go significantly higher over time?
Is your UWorld percentage supposed to go significantly higher over time? I would say on individual exams, yes, but on the average, no. The average will actually like. So like if I sit, like when I first started doing UWorld, I was averaging like what 30s and 40s. So like, let's say I was like here. So the thing is, I sat there for so long that like even when I started getting like 80s, 75s on my UWorld, it would just like slowly creep up my average like a snail. But like I realized that like I was getting like 30s, 40s, whatever it is, and then all of a sudden I was getting like 70s and 80s, and it's just like you sort of unlock the ability to understand UWorld better. So like that jump happens, but because of the amount of time that you spent like not really knowing what you're doing, your UWorld average is actually going to climb very slowly. I ended up with a 69% on my UWorld average. Nice. When should you be done? I used to say a month before your exam, but now because of the amount of content in AAMC, I'd say about 6 weeks before your exam. Just so that you have enough time to do everything. Why'd I throw that away? Does anyone here have like a sibling or like a cool cousin that they have who has been pregnant? Like they've seen them be pregnant? Is pregnancy brain a real thing?
No, it's my wife.
People always talk about like, you know, because I feel like Ramadan brain is a real thing.
Oh,
Ramadan brain is a real thing. Like,
I'm scoring lower on like
Although my wife it was a weird case. Like, she didn't have morning sickness. She still went on walks with me till like 8 months pregnant.
interesting.
Like, yeah, she barely felt. She was a tank.
Cool.
Monster.
I was 10 and a half pounds when I was born, so my mom was like
Yeah.
a balloon.
5 kilograms.
Look at that.
4.9 kilograms.
There you go.
Big boy.
Missense, we have nonsense mutations. We just said that this is an early stop codon. So you get a truncated protein. Protein's supposed to look like this. You get that. Right? That nonsense. Right? And then a frame shift mutation. A frame shift mutation is actually really toxic. This comes from deletion. Deletion of bases, a base or bases. What happens is that, let's say that you have a chain like this. I'm going to just do this. All right? A, U, 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 G, 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 C, C, C. And this produces number 1, number 2, number 3, and number 4. Then you get a deletion of a single base pair. And I'm going to block off the codons the way they come. I'm going to say A, U, U. I'm going to delete the third U. G, 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 C, C. Do you guys see how number 1 is preserved? This is completely different than that one. This is completely different than that one. This one's going to be completely different than that one. So after the deletion, you actually get a complete destruction of the DNA code. This is a horrendous mutation to have. This is really bad. Really, really bad. Right?
What's called frame shift?
A frame shift mutation. Because you shift the reading frame of the RNA.
I heard friendship, and I was like, wait, what?
Friendship?
Friendship mutation.
Yes, it sounds so good.
Sounds so much
I want that. There is another type of deletion that takes place, which you'll learn in medical school, and I just think it's important to bring up because you might see a passage about it every now and again. Cystic fibrosis. Cystic fibrosis is implicated by the, and this is another way to read codes, so forget all of this. This is a side conversation. It's noted by the delta F508 gene on the CTFR gene. Or sorry, is it CFTR? Let me look it up. So the cystic fibrosis transmembrane conductance regulator, CFTR, right? You get a delta F508. This is a phenylalanine in position 508 that gets deleted. But that's an entire codon deletion. That phenylalanine is just gone. And what that does is it actually leads to an aberrance of the chloride channel, which causes the symptoms of cystic fibrosis, which we don't need to get into. This isn't medical school, but you guys can look at that on your own time. So these are two sorts of what?
I want to watch this lecture again.
Why? What are you not understanding?
No, I'm not confused. I know this lecture.
Oh, okay.
I just,
Ramadan brain?
yeah. Everything looks so funny.
Don't worry. When I hear that you guys were at Sahur Fest instead of studying for the MCAT, I'm gonna come for you.
My mom said,
I'm gonna find you. By the way, I have such crazy opinions about that. We'll talk about it later as to not leave the white people out of the room. Also, cracking open a soda while I'm in the middle of talking about Ramadan is insane. That's crazy. That feels deliberate.
did you hear what she said?
Yes, I did. That feels deliberate.
My biochem passage was about cheese the other day.
Cheese?
Yeah, like the passage was about cheese.
Nice.
I was just saying, out of respect, I throated my food on the way here.
You did what to your food?
You heard me. You heard me. I filled the food, the water, and everything up here because I wasn't going to test your faith. But never mind.
Someone's got to do it, you know?
Yeah, right.
Benji's sitting there. He's like, "Damn, I could have my sandwich in here, man." Like,
Sorry.
personally, the thing is, I've been fasting every Ramadan since I was 10. It's like this is my 15th Ramadan or 16th. I forgot, 15th. I don't care.
It's like you're talking about being in medical school and I'm sitting here studying. It hurts.
Same thing with RNA transcription and synthesis of RNA polymerase. Helicase topoisomerase unwinds the DNA. RNA polymerase and DNA polymerase, and then you get transcription factors that help the RNA polymerase bind and move forward. It's specifically RNA polymerase 2, and you also have. Let's just do this. You have RNA polymerase 2, which is going to bind something known as the template strand of DNA.
Are the numbers important for the exam?
I don't know. Binds the template strand of DNA, and it specifically binds a portion of the template strand known as the TATA box. The TATA box is something that exists upstream of the gene that you're trying to transcribe. So that when the RNA polymerase attaches to the TATA box, you get a movement towards the gene and a transcription of the gene into mRNA. The numbers are listed in Kaplan, so let's go over them. You have RNA polymerase 1. You have RNA polymerase 2. And of course, after RNA polymerase 2 comes RNA polymerase. I wish. It's 3. I just love it when it's 1, 2, 5. This is localized to the nucleus and makes ribosomal RNA, rRNA. This is localized to the nucleus and makes mRNA. And this is localized to the nucleus and makes tRNA. And one more Whopper. Oh, my back. Here, Ricky's. I think the best fried chicken place nearby, I think I've had this conversation with you guys, is Mike's.
They opened up a new one near my
No way.
We're gonna go.
Yo, Mike's is so good. And the thing is, they make it fresh every single time you come in. Like, oh my
Been
god,
craving it.
so
She goes, it's 10 minutes from our house.
so good. Mike's is deep from Carrollton though.
How far is it from you? There's 2
I
locations.
think at 20 minutes.
Yeah.
Something like that. Like if I want to go to Epic, like that's a trip for me. Like Epic from Qalam, that's like far.
Yeah. You can either go to Lighthouse or you can go to Kalam.
Yeah. I haven't been to Lighthouse yet actually because we always have Sanctuary on Tuesday nights. Yeah. I've heard Lighthouse is nice though.
Yes.
Post-transcriptional processing. After we transcribe the DNA into RNA, we can have a level of processing that occurs in order to get the mRNA ready for synthesis of protein. You add a 5' GTP cap, more specifically a 7-methyl guanylate triphosphate. For simple terms, just say GTP. And a 3' polyadenosine tail. 3' poly A tail. And more specifically, in eukaryotes, we can do this. This is pretty cool. We have exons and we have introns. Exons get what? Exons get expressed, right? Exons are expressons, so they're the things that get expressed into proteins. Introns get spliced and cut out. Alternative splicing is the method by which you get this. You get that. And you also get this. Am I in frame?
Yes.
So one splice gives you exon 1, 2, and 4. The next splice gives you exon 1, 2, and 3. The next splice could give you 2, 3, and 4. The next splice could give you 1, 3, and 4. The next splice could give you 3 and 4. The next splice could give you 1 and 2. The next one could give you 2 and 3. So you can create all these different sort of combinations of the exons, and those will lead to the formation of different proteins. So one gene can control the production of all these different proteins by alternative splicing. Can bacteria do this? No, they can't. This is a complicated part of the chapter. Let's talk about the ribosome. So when the mRNA exits through the nuclear pores and encounters a ribosome, what's going to happen is that the ribosome has an aminoacyl site, an A site, a peptidyl site, a P site, and it has an exit site, an E site. you have three processes: initiation, elongation, and termination. Initiation starts when the small subunit of the ribosome binds the Shine-Delgrano sequence. I know. And this is in prokaryotes. So this is in bacteria. Or the 5' cap in eukaryotes. Remember that prokaryotes don't have that post-translational modification, so they can't have that 5' cap. tRNA, a charged tRNA molecule, binds the start codon and deposits methionine. And then after this, the large complex binds. And now it's known as the initiation complex. So you drop off that first amino acid, which is always going to be methionine, to start off the chain. And then you get the attachment of the larger subunit. What happens next is a 3-step cycle that is repeated for every single amino acid added after methionine. And this is the elongation process. So first, the A site holds incoming tRNA. The P site holds tRNA with the growing peptide chain. Let's look at a diagram of this real quick. This is the A site, the P site, and the E site. Here we have a tRNA with an amino acid on it. And here we have our tRNA, a tRNA, great tRNA molecule, by the way, if I must say so myself, with a chain on it. And what's going to happen as this shifts this way, this peptide chain, this amino acid will get added onto that, but in a fashion that the peptide chain actually transfers onto the next one that's coming this way. So there will always be a tRNA that's carrying the peptide chain from the last amino acid that was added. So this is going to attach to here, and this is going to break off from here. This guy's going to exit, and then we move on. You guys see how it's like an assembly line? It's just going to continue adding peptides, amino acids, until the peptide chain grows and eventually stops. And then everything that gets broken off from here, this guy now enters the E site and leaves. You guys get that? It's kind of difficult to visualize. An animation would be better. And then after the transfer, well, I guess we should say that the peptide bond is formed, and the polypeptide chain moves from P site to A site. So because the A site had the incoming tRNA, now it moves. And finally, the third step is that the uncharged, meaning no amino acid tRNA, escapes via E site. And the thing in the A site, which is now the polypeptide, moves to the P site. And we repeat. How are you guys feeling? Termination occurs when we have a stop codon moving into the A site. Stop codon enters the A site. Release factor binds and the ribosome dissociates. Hooray! The release factor leads to dissociation. After the formation of the protein will go on to either inside the cell or in the Golgi, and we have some post-translational processing, not post-transcriptional, but post-translational. This is stuff like phosphorylation, carboxylation, glycosylation, prenylation, blah, 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 blah. Who cares? Actually, I don't. You have to. There's a continued discussion about eukaryotes and operons. Just know the difference between inducible and repressible operons. Inducible operons are things like the lac operon, meaning that in the presence of lactose, you get the activation of the operon to transcribe genes that allow for lactose breakdown. Whereas repressible operons, like the trip operon, are constantly on, but once you have a level of tryptophan that binds the operon, or the repressor of the operon, eventually it gets turned off. So it's the difference between, I think one thing that people get wrong about operons is that it's the difference between constantly being on and constantly being off. Repressible operons are constantly off. Repressible operons are constantly on. And they can be turned off when you have enough of the thing you're trying to make. Inducible operons are constantly off. And they can be turned on when you have the thing that you need the genes and proteins to work for. Meaning you needed to break it down or organize it or do this or do that, whatever it may be. That's the end of chapter 7. Chapter 8. Oh, we're not done.
Oh.
No, we're just going to keep going.
Oh, no. Splice the video. How much time is left, do you think? Guesstimate.
30 minutes?
30 minutes?
Yeah. Well, you got to go?
No, but family time stuff,
Yeah, I mean, dude, the next, 2 chapters are really just, stuff we've already covered. So if you want to get out of here, then feel free.
Hey, man, I'm in it for the marathon.
This next chapter is literally just, membranes. We talked all about membranes and membrane potentials and this and that, whatever it may be. So.
Bro, I've been here since day one. I'm not going to...
All right.
Last real lecture.
Oh, for real. All right. Let's continue. Chapter 8. membranes. This is going to be a really dictated chapter. I don't think this chapter needs a lot of explanation all that much. Talk about the fluid mosaic model, how you have a semi-permeable phospholipid membrane, the two layers of phospholipids, hydrophobic tails, talked all about that, and polar heads on the outside. The general structure and function of membranes, so we have proteins, carbohydrates associated with the membranes to make membrane-bound proteins like glycolipid coat and blah, blah, and sometimes you have glycoproteins on the surface which act as signaling sort of domains, receptors, blah, 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 this is that. Also, in terms of like chapters that I used to take a lot of time describing when I taught this course for the first time versus now, I'm a lot more mature now. I've grown a lot as an MCAT tutor, and I realize that it's very stupid to teach everything to depth because you're not going to get asked many questions to depth about these things. So if there's something I'm just breezing through, it's because you're probably not going to get that many questions on it, or it's been elaborated so much in other lectures already. Okay. Lipids are the most abundant part of the phospholipid membrane. Obviously, they're phospholipids, right? And then you have fatty acids, triglycerols, that are the precursors to those phospholipids that can also exist in the membrane domain, right? Phospholipids, just to go over them one more time. If you have a triglyceride, and now this is fatty acyl, right? That's an acyl group because we have a double bond O and an unknown number of carbons. That's acyl. It would be acetyl if it was two of them. If you have this and you replace one of these with a phosphate, and I'm just going to draw it this way because we're used to seeing it this way, that's a phospholipid, right? And that's what we're saying when we draw something like that. That this is the circle and these are the two tails, right? These also serve structural roles and can play into secondary messenger pathways. And the phosphate may also be replaced if we need a positive or negative charge. So let's say that instead of the negative of the phosphate, we need a positive charge or a different type of negative charge or something different, or some sort of different molecule, whatever it may be. What do cholesterols do? This is actually pretty important. You remember how we always say cholesterol increases membrane fluidity, right? Cholesterol actually increases membrane fluidity in what? Yeah, when it's cold. So cholesterol increases fluidity in the cold and actually decreases fluidity in heat. Cool. Then in terms of proteins, we have embedded proteins, transmembrane proteins, integral proteins, and peripheral proteins. Transmembrane protein will look like that. An embedded protein will look like this. And a peripheral protein will look like this. Yeah, just associated with the membrane versus inside the membrane versus through the membrane. And both of these, transmembrane and embedded, are also known as integral proteins. Or integral. Carbohydrates help with signaling and recognition, right? So carbohydrates usually make up antigens. You guys remember chapter 8? General biology, immune system, MHC1, MHC2. Wow. What a time.
One goes as 8, 2 goes as 4.
There you go. Membrane receptors, usually transmembrane proteins. For example, ligand-gated ion channels. Let's take a moment to look at this. Let's talk about receptors in general. Where did the other one go? Ligand-gated ion channels. This means that X binds, and this leads to ion flow. Voltage-gated ion channels. Membrane potential reaches X threshold, and this leads to ion flow. Receptor tyrosine kinases. Ligand attachment leads to cross phosphorylation of tyrosine domains, which leads to signaling. This makes sense because tyrosine is one of the 3 amino acids that can be phosphorylated. What are the other 2? Serine and threonine. So what do you need in order to be phosphorylated? You need a OH group. And the last one that we're going to talk about is the G protein coupled receptor, which has that long list of steps that you guys need to know in order to, there's like the attachment and then the cyclic AMP and the blah blah and the this and that. Whatever. Yeah, I knew it at one point. The rest of this is stuff that we've covered already. Junctions. Cell-to-cell junctions allow for direct pathways of cellular communication, cellular adhesion, right? You have things like membrane transport, so passive transport, active transport, we've talked about that before. Passive transport doesn't use energy, active transport does. Osmosis, you guys know all about osmosis. If you put something in a hypertonic solution versus a hypotonic solution versus an isotonic solution. I have my whole lecture on fluids, which is part of our chapter 10 metabolism thing. We also have some supplemental videos on fluids if you guys want to check that out. I just don't want to waste your time. Okay, this is interesting. So what's the difference between facilitated diffusion and active diffusion? Or active transport? Passive diffusion versus facilitated diffusion versus active transport. Facilitated diffusion still doesn't use energy, but it uses carrier proteins or channels, right? So they open or close depending on how much of the thing you want to let in. So if we're looking at the differences here, we have something like simple diffusion. A leads to B. This goes from here to here. Facilitated diffusion means that A wants to go here, but it needs something to help. And then it goes to B. Active means that A wants to go to B, but in order to do so, you need a protein and some sort of energy transfer. And that would be something like a sodium-potassium pump. Now, that specifically is primary active transport. Secondary active transport is a little bit different. It still uses energy, but it harnesses energy in a different way. Secondary active transport is when the movement of a molecule is powered by another molecule moving along its concentration gradient. So let's take a look at this. You want to get into the club. and there's a bunch of semicircular molecules in the club, and you are this crescent-shaped molecule, right? You want to get in, but you know there's a ton of crescent-shaped molecules in here. So in order to get in, you need energy to come and help you. But instead of using energy, we know that it just looks like C's and D's. Okay, let's try this again. You are a dot. There's a lot of dots inside of the club. And you want to come in as a triangle. There's a ton of triangles in this club. And you're trying to get in. You need some form of energy. But if there's very few dots out here, what you can do instead is take the dots and move them out, which will release energy because of the fact that that's what that wants to do. And that release of energy will power you to move in. That's called secondary active transport. Because we're trading off the comfort of taking the dots out with the discomfort of moving the triangles in. Make sense?
Yeah.
Do you guys remember exo and endocytosis? In this chapter, they make a specific distinction where you have endocytosis, also called penocytosis. Penocytosis is endocytosis but with fluids. That's it.
I know it's just an example on the board, but if all the dots are out and it's full of triangles, there's no movement anymore, which is cellular death, isn't it?
Sorry?
I know this is just an example that you're putting on the board, but wouldn't it make sense to remove a triangle to put another triangle in? But you're removing a dot. Eventually, all
Yes.
the dots are gonna be gone.
So the thing is, like not all, right? So your question is like, this is just an example of like, I'm talking about like there's, for each of those probably 10,000,
Okay, got it,
right?
got it.
So we're taking out like 10,000 dots to move in 1,000 triangles.
I'm reading too much into it.
You know what I mean? So like the metaphor isn't perfect. You know what I mean? And it's also actually difficult to find an example of where and how that happens. So, you know. Have you guys seen that? Never mind. Okay. I think that's it. Okay, we are going to do chapter. Let me organize my thoughts before I end the video. The people at home need to watch me organize my thoughts. They have to. They must, huh? I know I'm not. I'm not supposed to be. My voice is in frame, and that's all that matters. Chapter 3 is honestly like, yeah. All right, I'll see you guys in the next one. Bye. Okay, let's continue on to chapter 3. Chapter 3 really is just like super quick, almost as quick as chapter 8, which was membranes, which we just did like, how many seconds ago was that? Like 15? Yeah. Okay, so chapter 3 goes very, very fast. If you can't tell, all of these chapters mean very little to me. And they should mean very little to you as well. Because that's all it is. Chapter 3 is non-enzymatic protein functions. You're basically only going to be asked about enzymes, right? So this chapter is very low yield. We have some structural proteins. Let's go over the structural proteins. Collagen, elastin, keratin, actin, and tubulin. Collagen, is important in connective tissue and is important for strength and flexibility. Anyone know what disease interrupts collagen formation? Military should know. Scurvy.
Wait, scurvy?
Scurvy.
Oh,
Vitamin C
vitamin
deficiency.
C, right?
Vitamin C deficiency. Yep. Why your teeth fall out and you start bleeding? Because you don't have enough collagen to maintain all those like binding pockets in the body.
Forces of the Navy should know that.
Elastin. Its job is to stretch and recoil like a spring. So elastin would be very important in the walls of arteries. Elastic. They want to be able to come back to where they are. Keratin. These are intermediate filament proteins. What does that mean? I have absolutely no idea. Mechanical integrity. Whatever that means. This is also found in hair and nails. What's the important bond in keratin? Disulfide bonds. Very good. But perms, yeah. Someone asked me to get a perm one time. I told them something I can't say right now. Or I should never say ever again to anyone. No, like the thing is, okay, I'm going to get a lot of hate for this, but I don't care. If you are a fully grown frontal lobe developed man and you are walking around with a perm and your eyebrows done, I think of you as lesser. I think of you as less of a secure person. Like the eyebrows done thing is personal because like religiously Muslim men are not allowed to do their eyebrows unless you have a unibrow in which you're able to remove the hair right between here. But shaving and shaping of the eyebrows is forbidden for men and women. You're actually not allowed to do that. People will ask why. Just 'cause.
I thought women were allowed to shape their eyebrows.
Women cannot shape their eyebrows. It's not allowed. But for men, it's even more so not allowed.
Because it's a feminine trait.
Yeah. Women naturally have like thinner eyebrows than men do. So like the idea of men like thinning out their eyebrows is like a feminine thing. You know what I mean? And then the perm, like about the perm, it just doesn't look good. It's just ugly. It's really, really ugly, especially on men, and especially on men who already have very thick, curly hair. I have relatively wavy, curly hair when I grow it out. You can't tell now, but if you've seen my videos with my long hair, it's not straight. My hair is definitely not straight. And I was going to make a joke, and I think you know the joke I was going to make, but I'm not going to make it right now. So yeah, don't get perms. Don't do that. I don't want to see that, and I promise you no one else does either.
Oh, you gotta get the perm, the mango.
No. You have to get the perm with the strawberry matcha, light ice, the vape in the pocket that you're hiding under your sweater during Ramadan so that no one sees you, and then you get the eyebrow slit. That's the one. That's the one.
So when you said eyebrow shaping, do you mean like slit or like?
No, shaping them. Like getting them like shaped and things. I've never touched my eyebrows before.
No, not for men.
Yeah.
I've never heard that.
Yeah, no, women aren't allowed to do it either.
Interesting.
Unless it's insanely like masculine. It's
Yeah.
a problem.
There's always like an allowance for things. It's just like if it's out of the ordinary. Like for example, interestingly enough, there is an opinion. There's a scholarly opinion in Islam, and this is like not joking at all. This is a very serious discussion. There's a scholarly opinion in Islam that if a woman has a completely flat chest, like so flat that it looks like a man's chest, She is allowed to get implants in order to appear feminine, because that is something that is out of the physiologic norm. You know what I mean? Because at that point, it's something that is like different. It's something that is outside of the ordinary.
It's to fix abnormality.
Exactly. So like plastic surgery in general, like as a general clause, is not exactly like the most openly accepted thing in our religious principle. But if it's something that is outside of the norm and it's a defect that you're fixing, then there are different rules that apply to that.
I mean, they used to get like perception something about handles.
Like restoring.
Restoring something to which it once was. That's also something that people...
Given birth, or people who have
Another one. If a woman has, in the complete opposite realm, if a woman has very large breasts and it causes her pain, she's allowed to get a reduction. Actin. These are proteins implicated in muscle contraction. Long story short, don't get a perm. And if you were thinking of getting a perm, fear God. And also, just out of respect for the people around you who have to see that, don't do it. It's so ugly. It's so ugly. It looks so bad. Yes.
Could you read the actin?
Oh, it's proteins implicated in muscle contraction.
Who with the perm hurt you?
Huh?
Who with the perm
No, it's just every time I see one, it just pisses me off. Like it makes me so mad. Like people have their thing, like that just is like it hurts my soul. Tubulin. These make up microtubules. Motor proteins like myosin, kinesin, dynein. Myosin is the one that actually allows for muscle contraction when it pulls on the actin filaments, right? We talked about that whole thing, chapter 11 of general biology, the myosin power stroke, where you get like the myosin in the uncocked position versus the cocked position versus the ATP versus the ATP versus the movement versus the blah, 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 this is that, whatever. Okay, fine. Kinesin and dynein. Dynein, remember, dives into the cell, so it moves towards the cell center. And kinesin moves the opposite way, right? We talked about this already. Binding proteins, things like hemoglobin can bind to oxygen, right? Cell adhesion molecules, you have cadherins, integrins, selectins, right? Selectins are important for white blood cells entering into different endothelial beds. Immunoglobulins, talked about that, all of it in the immune system. Biosignaling, ion channels, we talked about that, all in biology as well. Enzyme-linked receptors, talked about that, all in endocrinology and enzymes. G-colored protein receptors, beat that one with a dead horse. Actually, I didn't, but you did, because I don't want to teach it again. Then we talk about protein electrophoresis and PAGE and SDS PAGE and isoelectrofocusing and chromatography and column chromatography and protein structure and amino acid composition and activity analysis and all of that stuff. And every single one of those things I just mentioned about protein analysis is mentioned inside of my lab techniques video. That's the end of the chapter. Thank you guys so much for watching. Let's do chapter 12.